And good morning. It's Dr. Roddenberry coming to you live from Willow Springs, North Carolina. Good morning, everybody. I see we already have some people in our audience today. If you're in the audience, go ahead and check in the chat bar. Let me see you're here. Probably Luis is here, maybe. Martine is here. Good morning. Good to see you. Thanks for coming today. Anybody else in the chat bar right now? This is the last chat of the week, the last lecture of the week. And then remember, we have the discussion board that's due tonight at 11:59 p.m. I'd like to thank the 24 people that came out to the webinar last night. We had a good time and talked a little bit about the modularity of vision and the difference in levels of processing of different kinds of visual information. And I had some cool videos to share. But don't forget, your discussion board is due tonight, and homework number 3 is due tomorrow night at 11:59 p.m. Good morning, Andrew. Thanks for coming in. Martine, thanks for coming in. If anyone else is in the audience, go ahead and type in the chat bar, but thank you very much for coming. So today we're going to be talking about the second half of Chapter 5. We're going to talk a little bit about the other senses. So if you would give me about a half an hour, 40 minutes of your time, and we'll go through and talk a little bit about the other sensory processing systems in your nervous system. Good morning, Heaven. Thanks for joining the broadcast. Looks like we've got a couple of other people. Just to remind you, don't forget to do the discussion board that's due tonight at 11:59 p.m. And don't forget Chapter 3 quiz, which is due tomorrow night at 11:59 p.m. I'll be sending you reminders around 6 o'clock today, just to keep it from slipping your mind. So let's go back and revisit the chart that we talked about yesterday. Remember, there are 5 primary sensory cortices that I want you to be familiar with in the cerebral cortex. Remember, the gustatory and the olfactory, taste and smell, are in the frontal lobe, The sensory cortex, the primary somatosensory cortex, is in the back part of the parietal lobe, All right, and down below those, the auditory cortex is in your temporal lobe. And then I want you to know that the primary visual cortex is way, way, way in the back of your brain in the occipital lobe. Okay? Now, excuse me. Yesterday in vision, I talked about the importance of the thalamus. I suggested to you that the visual information comes back through the thalamus and splits up into a couple of different ways. So one part goes to your superior colliculus and coordinates your hands with your other senses, and the other provides a straight direction back to the primary visual cortex. But the thalamus is a very important part of your brain for helping your senses coordinate with another. So what I do want you to know is that all of your conscious sensory experiences, except smell, are processed through the thalamus. I guess smell might be so early that it developed in an evolutionary perspective before the thalamus, so it doesn't go, the olfactory tracts do not go through the thalamus. But all of the other senses are processed through the structure in your midbrain called the thalamus. Okay? Now, remember, and I hit this a couple of times before. I'm going to hit it. I talked about it last night, and I'm going to hit it again. Remember, not every spot in your brain is given for a specific predetermined function. You do have areas in your brain called association cortices where these senses can work with other senses to create higher level perceptions. And last night we looked at some videos of some people who couldn't recognize car keys, glasses, or a combination lock. So these sensory experiences that we have need a place where they can go and combine themselves to make final full perceptions that we can then consciously recognize. This occurs in the association cortices, right? So I want you to know the lobes where each of these senses are. I want you to know that all of them except for smell go through the thalamus, and then I want you to know that association cortices are there to help put these sensory experiences together. Okay, so let's talk a little bit about the mechanoreceptors in your ear. So you have rods and cone cells in your eye, and their job is to transduce light energy, photons, packets of light energy, into patterns of neural activity. Well, in hearing, what we have are hair cells, and these hair cells are impacted by sound pressure created by sound waves. So you have little hairs in your inner ear in this structure called a cochlea, and as sound comes in your ears, it causes the liquid that these hair cells are floating in to move, and it causes these hair cells to move, and that's what transduces sound energy into patterns of neural activity. Let's take a look here at the ear picture that we have moving left to right. You see we have the outer ear, we have the middle ear, and we have the inner ear. In the outer ear, you're going to see the pinna, the auditory canal, and the eardrum right there in the outer ear. Your middle ear is going to consist of some bones that then connect to this inner ear structure called a cochlea. And you'll see that in the second part of this picture. So, what happens is these sound waves come in and are captured by these outer structure of your ear, the pinna, and funneled into your ear where it makes this thing called an eardrum vibrate. And that's right there at the end of your outer ear and kind of transitioning into your middle ear. This eardrum vibrates, and there are three bones that are connected, that connect this eardrum with another drum-like area called the oval window. So you have three little bones. And what happens is when the eardrum vibrates, it causes these bones in your ear, the hammer, the anvil, and the stirrup. They're the smallest bones in your body. They're right here in your middle ear. It causes them to vibrate, and then they cause this oval window to vibrate. Now, why not just have the sound waves vibrate your eardrum and then have that move the hair cells? Well, what happens is with these three little bones connecting to the eardrum to the oval window in your inner ear, it magnifies the vibration. So these three little bones and their connection to the oval window magnifies the power of the sound waves, which makes it easier for you to perceive these sounds. Now, if you go into the middle picture where it talks about sensation, what you see is your cochlea, which is this fluid-filled structure in your inner ear that's actually wrapped. And you'll see it's kind of circular in picture 1, and they've unrolled it a little bit in picture 2. And what you see is that this structure, going along this structure, you have this basilar membrane with these hair cells growing out of it. And there's fluid floating all around these hair cells. Now what happens is when the inner ear bones vibrate and cause that oval window to start moving, it causes the fluid in the cochlea to begin moving. And that fluid causes the hair cells on your basilar membrane to move. And that causes the transduction of sound energy, which is external energy, into a pattern of neural impulses that then leave your auditory cortex, travel down through your thalamus and into your, that travel from your inner ear through your thalamus to your primary auditory cortex. All right. So looking at this picture here, the things that I want you to know: the oval window vibrates, and that transmits sound energy to your inner ear through these three bones. These three bones cause another window, the oval window, to vibrate, sort of magnifies the sound, the value of the sound waves, and it causes this fluid in the inner ear to vibrate, moving these hair cells. And the movement of these hair cells is what's transduced into patterns of action potentials that then travel out your auditory nerve through your thalamus and into your primary auditory cortex. Okay? Now, there's one other sense that I want to talk about right now, and that's your sense of balance, because that is all. Okay, I am sorry. Looks like I lost internet for just a second, but I'm back. All right. So, there's another sense I do want to take a minute to talk about. If you look in picture 1, you'll see these structures called the semicircular canals. It is 3 canals that are oriented in different directions, and they're filled with fluid and these same hair cells. And what happens is, as your head turns sideways to this way, it causes the fluid in these semicircular canals to flow over these hair cells, and the movement of this fluid over these three circular canals that are oriented in different directions, this information is then sent to your cerebellum and creates a sense of balance for you. So your inner ear is responsible for hearing and for balance. You have hair cells in both your cochlea and your semicircular canal. The sound waves that come in your ear cause the hair cells in your cochlea to move back and forth, creating a sense of sound. On the other hand, the fluid-filled semicircular canals have hair cells that are moved by fluid that's floating around in your head as you move your head in different directions. And it's this fluid floating around, moving the hair cells in your semicircular canal, that is then transmitted to your cerebellum and gives you a sense of balance. So really, you have hair cells that are responsible for 2 senses in your inner ear: your sense of balance and your sense of hearing. Okay, any questions so far on your sense of hearing? Now let's talk a little bit more about the cochlea, this fluid-filled rolled-up tube containing the basilar membrane and the hair cells. This is where the qualities of sound are transduced. Okay, so the basic sound qualities that we perceive are pitch, whether or not the sound is high or low, and whether or not the sound is quiet or whether it's loud, right? Or whether it's over here or whether or not it's coming from over here. So if you think about it, our basic sound qualities that the ear is responsible for perceiving is pitch, loudness, and the localization of sound. Okay, so there are 2 sort of competing theories on how pitch, high versus low, is created, but the evidence seems to suggest this thing called place coding. So you have hairs all along the length of this rolled up cochlea. And what they found is that when the hair cells vibrate at the beginning of the cochlea, produces a really high sound pitch, and when the hairs in the back of the cochlea vibrate, it produces a low sound pitch. So one of the theories that appears to explain how we determine when things are high pitched or low pitched is on which hairs in the cochlea actually vibrate. And that information is transmitted to your occipital cortex, which compares the vibration of the hairs in the different parts of the cochlea. And I am just not sure why I am disconnecting this morning. I do apologize for that. And so place coding explains how we perceive high and low pitch. All right, now Loudness is generally determined by the sound wave amplitude. So if you look at these tubes up here, frequency is how quickly the waves move, how quickly it goes from trough to trough or peak to peak. And amplitude is how high up and down the wave goes. And loudness is generally determined by the sound wave amplitude. Higher wave sound amplitude is actually perceived by your brain as a louder sound. So these bigger amplitude waves are going to create more pressure in the ear, which you are going to perceive as loudness. Now, just like we said, you have two eyes that are each looking at something from a slightly different perspective. You've got two ears that are receiving sound at a slightly different perspective. Now, those of you who came to class last night were shocked to see Ben Underwood, and he was actually able to use his ears like sonars to actually create a visual picture just by sounds. So the one thing you need to remember is that your ears are pretty good with timing of sounds. And so what happens is you've got your two ears, and when something makes a sound, depending upon where it comes from, the information reaches one ear before it reaches the other. And your brain learns how to interpret the difference in timing between when the two sounds are recorded in your brain as a measure of location. So you'll see this fellow down here right below me. He's got his head turned this way, and the sound is coming from over here. So it means it's reaching this ear first, and then it's reaching this ear second. And your brain interprets that to give you an idea of localization, where sound is coming from. Good morning, Marcella. Thanks for coming in. Good morning, Salem. Thanks for joining us. We're just finishing up our discussion of the ear. So the things I want you to know: cochlea, hair cells, place coding, and how sound is localized. Those are some of the most important questions that I'm going to ask you on the exam. Now, quickly, just as a memory check, which all of the senses except this one go through the thalamus. Which sense is not processed through the thalamus? I'm going to go ahead and leave that open in the chat bar. Hopefully somebody can type in the correct answer and let us all know. Okay. Now, taste and smell are what we call chemoreception. These two sensory processes are responding to chemicals. And in both of these cases, we have chemicals, either odorants that floating chemicals that float in through your nose, or more solid chemicals that are in foods that you eat. And these molecules fit into receptor sites perfectly and activate these receptor sites. Kind of reminds me in a way of the synaptic receptor sites. We talked about proteins that are fit by molecules perfectly that cause them to activate. You can think of chemoreception along the same lines. Your chemoreceptors are looking for specifically shaped molecules to fit into them to activate them so that you can experience the sense of taste or the experience of smell. Very good, Mariah. Smell is the only of the five senses that is not processed through the thalamus. Olfactory, fantastic. Great, great, great. Okay, I would agree. Now, taste receptors. So your taste receptors are in your taste buds located on your tongue. We have about 8,000 to 10,000 taste buds in our mouth. And if you see the picture way down in the bottom corner, you see that tongue coming out, and you see all of those taste buds located. And the taste buds start on the tip of your tongue and actually go down towards the back of your throat. And these receptor cells transduce chemical stimulation into action potentials. Okay, this then travels along the bottom of your tongue, your facial nerve, comes up through your thalamus, and then it goes to your goes to your gustatory cortex. Okay, now it turns out if you look, our tongue has different types of taste receptors that report to different parts of our gustatory cortex. So in your gustatory cortex, you have an area that gives you the sensation of sweet. In your gustatory cortex, you have an area that gives, when stimulated, gives you the taste of salty. When you have another area in your gustatory cortex, when it's stimulated, it gives you the taste of bitter or of sour or of this meaty, tasty, salty goodness, beef stew taste we call umami. And so the deal is, on your tongue you have different receptors located in different areas that connect with different parts of your brain. So you have taste receptors that are respond to sugars, so glucose, sucrose, fructose, and these taste cells, receptor cells, are located mostly in the front part of your tongue. Okay, you also have receptor cells that are that are stimulated by hydrogen ions, and they're located on the sides of your tongue, and they send a message to this part of your gustatory cortex that gives you the perception of sour. And then you have a part of your taste receptors that respond to salt molecules, potassiums and sodiums, and these taste receptors go to the part of your gustatory cortex that gives you the taste of salty. We've also got taste receptors that respond to glutamate, which is sort of that protein meaty flavor that we all love. If you've ever eaten a steak or a pot roast or smelled sort of the taste of mushrooms and pot roast cooking together, that meaty, wonderful sort of home cooked taste smell, that's the smell of glutamate, which gives you that umami taste. Now, we also have taste buds, our taste receptors in the back of our tongue that respond to lots of different bitter molecules. And what you're going to find is that the molecules that are usually toxic also okay, you know what, I'm going to go ahead and, while I'm looking at this, let me do a oopla. Hold on just a second. My kid's not playing on the internet, so I just wanted to check and see. What does my speed test say? My speed test says my numbers are all right. Download speed in the 80s. That's interesting. All right, I apologize for... Okay. You know what? I'm going to go with my hard connection starting tomorrow. Okay, we've got this back together. So we've got these taste receptors. Some people think that the taste of bitter is a protective mechanism to keep animals, humans, from accidentally poisoning themselves. So if things that are typically toxic also have a characteristic molecular shape, you can think of your bitter taste buds as a way to protect you from eating something that is dangerous, either spoiled or poisonous. And so these are the 5 main tastes. They go back to your brain, and as I suggested to you with vision, the final sensory perceptual experience depends upon a combination of factors. So we can have all kinds of complex tastes based upon how we combine these different molecular senses. And then, of course, there are memories that go with tastes, and memories also contribute into our final sensory experience. So something you eat may create a memory in your brain, may rejuvenate a memory of some event that's happened in your past. And so just like vision is a complex perception based upon your basic senses and your memory and other parts of your cortex, we can come up with the rich, complex sensory experiences for taste that we do have for vision. Oh, hello, Imani. Good to see you. Thanks for stopping by. We're just talking a little bit about sensory perception. Imani was a student of mine last semester. As you'll see, students always like to stop by and say hello, and I'm always glad to see them, especially the good ones like Imani. Okay, now, chemoreceptors. Now, chemoreceptors are also your olfactory receptors are also chemoreceptors as well. They respond to chemical molecules just like taste does, except they respond to floating chemicals called odorants that come in, and they come up here and they go on this sort of they stimulate these sensory receptors that are in our nasal cavities. So if you look right here, we have the odorants, whatever that smell of breaking bread, it comes into our nasal passage, and then what we have are these sensory receptors that are in the top of our nasal passage that are coming down. You'll see they're coming down from the olfactory bulb right there. You'll see these little nerve fibers, and these little chemoreceptors, olfactory receptors that are located in a mucus layer, they respond to chemical stimulation. Now, your book's going to say that there are 350 different types of odorant receptors. Now, we only had 5 tastes. We only had rods and cones, right? But we've got 350 different type of odorant receptors in our nose. And so what happens is these odorant receptors are activated by different molecules, and this information then travels to your olfactory cortex, and your olfactory cortex builds complex smells by putting together patterns of neural activity, neural stimulation that come from these 350 different types of odorant receptors. Now, the odorant traveling to these olfactory receptor cells and causing them to activate, that's the part that we call transduction. Taking these odorant molecules and creating patterns of neural activity from them is called olfactory transduction. And so these receptor cells, they come from the olfactory bulb, and the olfactory bulb is where your receptor cells are going to synapse with your sensory neurons that are traveling along your olfactory nerve. Okay, so you've got these little receptor cells that go from your olfactory bulb into the top part of your nasal cavity in this mucus, and those are your receptor cells. They then synapse with a different axon that carries from the olfactory receptor to the olfactory cortex. Remember, this is the only this is the only nerve that does not go through the thalamus. And who knows why, but one theory might be from an evolutionary standpoint, this sense of smell developed so early that it preceded the development of the thalamus. Now, one thing I do want you to know, although it doesn't go through the thalamus, the olfactory nerve travels through parts of the limbic system. Remember, the limbic system include the hippocampus and the amygdala, along with this entorhinal cortex. Don't worry about that. All I want you to know for this book is the hippocampus and the amygdala. Your olfactory nerve is going to connect to these two parts of your memory system, and this is a lot of times why we think a lot of times that certain smells can evoke certain memories. So maybe there's a certain food you associate with Christmas. Maybe there's a certain food that makes you think of grandma or makes you think of birthday parties. And it turns out that that smells are really good at evoking memories in people. Okay? Now, let's talk briefly about the sense of touch, recording pressure, pain, and heat. Now, think about this. Your visual receptor cells are located right here in your eyes. Your hair cells are located only in your inner ear. Your taste receptors are located only on your tongue, and your olfactory receptors are located only in your nose. But your sense of pressure, pain, and heat actually comes from all over your body. So the sense of touch, the proprioception, the sense of pain, the sense of heat is actually something that is perceived all over your body. And in fact, I'm even going to suggest not only is it all over the outside of your body, but especially with your heat receptors, they're going to be very important in measuring these sensations in your internal organs that you're not even aware of. So part of your brain, your hypothalamus, is responsible for managing the heat, the temperature in your body. Well, in order to manage the temperature in your body, your hypothalamus has to be able to sense the temperature inside your body. And so the sense of touch, the sense of pressure, the sense of itch, the sense of pain, the sense of being hot or cold are collected by receptors that cover your body outside and inside. So that's how touch is different from these other senses. Now we're going to talk specifically about proprioception, the recording of touch and pressure, and we're going to talk about these mechanoreceptors, and we're going to talk about the somatosensory cortex, which is located in your parietal lobe, right behind your frontal lobe. All right, and so these multiple types of mechanoreceptor cells in your body record different types of force: vibration, stretching, heat, and damage. So when you stretch your arm out, that affects these stretch receptors that tell you that your arm is now stretched in an outward direction. All right, you have vibration receptors, which can tell you when a little creature is walking on your arm by feeling the vibration of the skin. You have hair receptors that respond to when things move the hairs on your arm as well. Okay, you have itch receptors and you have pain receptors that can tell you when your structural integrity of your body has been violated by a knife or some sort of other dangerous thing. All right. And your pain receptors are even responsible to different chemicals. So like when an animal bites you and stings you, the poison in that animal's venom is going to create a chemical reaction in your skin that you're going to perceive as pain. So we have all of these different kinds of receptors located in our skin. Now, I'm not going to go ahead and go through each of these different receptors and describe the specific processes that these use. This is not appropriate for an introductory psychology class. This is more of a biopsychology question, or a question if you were to take a sensation and perception class, an advanced class. So I'm just going to mention a couple of broad things about how these sensory receptors work. Now, one of the things I want you to look at over here is this picture of the primary somatosensory cortex. This is, if you'll see the pictures of that human being right there, this is sometimes referred to as the motor homunculus. So homunculus is a monster. And so if you draw a picture of the monster and make its body parts as big as their space representational space is on the cortex, what you're going to find is that the body parts, as they are represented on the somatosensory cortex, are not similar to the size of the body parts in reality. So you have much more space in your somatosensory cortex for feeling your face and your lips and your eyes than you do for your entire leg. But your leg is so much bigger, but you have more sensory space on your brain for your face. And what we know is that the organizing principle in the sensory cortex is that the size of the area in the sensory cortex is dependent upon the complexity of the body part's action, not its size. So legs do one basic thing. They move up and down piston-like fashion and help us walk or run, okay? However, our mouth and our face does all kinds of crazy things. It creates emotions. I'm sad. It talks. It kisses. It does all kinds of crazy things. It eats. So our mouth feels and does a lot of complex behaviors. So we need more space to perceive these different parts of our brain. Now we have these dermatomes, so each part of your body is represented on your brain, and you have like a neuron which is responsible for perceiving senses in that different parts of your body. And what you're going to notice is that the dermatomes, the areas of responsibility for neurons, for sensory neurons, are much smaller in your face area, but these dermatomes are much bigger on like your back. So you don't have as much feeling on your back as you do on your face. So we have warm receptors and cold receptors, which basically measure your body temperature, interior and outside, compared to what the hypothalamus would like for you to be. So your hypothalamus has a set point, 98.6, and these warm and cold receptors respond to the temperature, evaluate the temperature on your skin, your surface temperature, and then we often talk about this thing called your core temperature, right? And we say your surface temperature can change. If you're outside, your skin can get cold and maybe your temperature can drop down. Your surface temperature can drop down to 65 or 70 degrees. But you also have temperature sensors in the interior of your body that give you a temperature of your core. And your core temperature has to be much closer to 98.6 than your surface temperature. And so we have warm and cold receptors, which evaluate the heat level inside and outside your body at any given time. We have 5 different types of pressure receptors: hair receptors, vibration receptors, fast pressure, and light pressure, and stretching. Now, the weird thing is our body, along with our balance, which way our head is going, our cerebellum also gets feedback from whether or not body parts are stretched out or close, where body parts are relative to one another. And so we get this kinesthetic sense that allows us to tell where in the world our body parts are. So I don't even need to look. I can tell you that my feet are down here. I don't even have to look. I can tell you that my left elbow is right over here because I have a set of kinesthetic sense. Now, there are people with damage to their brain, and what you'll see is that some people, when they're not paying attention to body parts, those body parts will start floating. So I have seen pictures of stroke victims that when they're looking at their right arm, it doesn't move anywhere. But as soon as they quit paying attention to it, they lose kinesthetic sense of where that body part is, and it causes that body part to move because the cerebellum can't control it anymore. All right. Now, we also have nociception, which is the perception of pain. The perception of pain is actually dependent upon 2 things. You do have chemical stimulation. So when we are hurt, our body releases chemicals. Whenever poison comes into our system, this poison is contained by chemicals. And so part of our pain response is responding to chemical changes inside the body, and we perceive that as pain. But we also perceive structural damage as pain too. So that knife is going to come in, and it's going to, as that knife enters your body, it's going to cut and damage and aggravate these nocifibers that we have in our brain. So structural damage and chemical changes are both going to be perceived by your body as pain. Now we have two kinds of fibers that your book's going to talk about: fast fibers and slow fibers. Now, I haven't mentioned this yet, but the thicker your axons, the faster information is transmitted from your body part to your brain. So actually, fatter axons travel quicker, action potentials travel quicker on fatter axons than they do on skinnier axons. When it comes to pain, you actually have two different fiber types. You have these big, fat, fast fibers that are thicker and myelinated, and they transmit acute pain. So when you step on a tack or you step on something sharp, that sharp information is sent to your brain super quickly so that you can move away from whatever that noxious thing is. If you stick your finger in somewhere and you touch a knife or electricity and it sends a sharp pain, your body needs to respond as quickly as possible. And so that information is transmitted on your fast fibers. On the other hand, you folks have all had dull aching pain. Maybe your knee hurts from what you did yesterday, or your back is hurting from lifting too many weights yesterday, or whatnot. These slow, dull, aching pains are actually transmitted on a different nociceptor fiber. And these are slow fibers. These are thinner and unmyelinated, and they transmit dull, aching pains. Okay? So, those are the 5 senses that I wanted to cover today. I wanted you again to know about the difference, where the different senses are processed. Your sensory, your gustatory and olfactory cortex are located in your frontal lobe. Your sensory cortex that feels your body is in your parietal lobe. Your auditory cortex is in your temporal lobe, and your visual cortex is in your occipital lobe. Now, these areas do work together, and they actually also, even individual areas within these cortices, build complex perceptions through the use of these association areas that allow parts of these cortices to communicate with one another and allow these cortices to communicate with each other, so that sound and sight and taste all can be used to build complex sensory perceptions. I do want you to know these different structures. I want you to know hair cells are in your cochlea, and I want you to know the cochlea and the semicircular canal, and these hair cells give us the sense of loudness, of pitch, and of balance. Okay, I want you to know that the fact that we have 2 different ears separated from one another our brain uses that to interpret the location of sound. I want you to know about the 5 different tastes on the tongue, the taste receptors, and how that information travels to the brain. I want you to know about the olfactory receptors and the response, and the fact that they respond to floating chemicals we call odorants. I want you to know that we have 350 different types of odorant receptors, and that smell is the only sense that doesn't go through the thalamus, but it does go to your memory system, which is why smells cause us to experience memories. I do want you to know that our motor cortex is organized such that our sensory cortex is organized such that the amount of sensory space you have on the cortex depends on the complexity of the body part and not necessarily its size. And I do want you to know that that we have temperature sensors, as well as pressure sensors, as well as stretch receptors, as well as pain receptors, and that all of these receptors are scattered throughout our body with touch, and they allow us to feel things, to feel warmth, to judge location of different body parts, and to feel when those body parts are injured. Does anybody have any question? Marcella's talking about reflexes. What do you mean by reflexes? And Heaven, I see you retracted a message. If you had a question and you felt like it was stupid, it's not. So please feel free to type that question back in again if you want. That's really all I wanted to talk about with the senses. Just to go back to yesterday, I am interested in you knowing the basic sensory pathway for the eyes, what the optic chiasm does, what the superior colliculus does, what the role of the thalamus is in connecting the eyes to the other visual senses. So hopefully everybody's got a basic idea of how our body takes in information from the external world and transduces it into patterns of electrical activity that we then learn how to interpret to decide what's going on in and around our body. Do we have any questions at this point? I am sorry for the disturbances in the broadcast today. I do not know what's going on, but I will look into it after class and figure out what the problem is. Any questions? All right, folks, I'm going to go ahead and log off now. Have a great day. If you have any questions, don't hesitate to send me a text or an email. Have a good day. Take care.